So chapter 12 deals with DNA and RNA, and 12.1 is going to deal with DNA. 12.2 is also going to deal with DNA, and then we'll get into RNA in 12.3. So the first thing we're going to talk about is the basis for all the experiments that were done to determine the structure and function of DNA. The first one is Griffiths Experiment, where he took disease-causing and harmless bacteria and injected both into a mouse, as we see in those first two columns. And the disease-causing bacteria, the smooth colonies, kill the mouse, and the harmless bacteria, the mouse is fine. And then he takes the harmful bacteria and uses a flame to heat kill the bacteria, injects them in the mouse lives, and when he goes to grow these heat-killed harmful bacteria, he gets no growth. Now he mixes the two that had the mouse survive, the heat-killed smooth colonies, the heat-killed disease-causing, and the harmless. And when he mixes them together, the mouse gets pneumonia and dies, and they grow into smooth colonies. So what he's seeing here is he's seeing transformation. Something was transferred between them. So transformation is the changing of something into something else. Now, Griffiths determined that there must be some factor that was transferred between the heat-killed cells and the live cells, and he hypothesized that the heat-killed bacteria had a gene that was transferred that could change the harmless bacteria into harmful bacteria. Okay, so there was some gene that was transferred between the two that caused that transformation. Now, Avery was another scientist who repeated Griffiths' work in an attempt to see which gene carried the information to turn the harmless into harmful bacteria. Avery and other scientists discovered that DNA is the nucleic acid that stores and transmits the genetic information from one generation to the next. Now this last term, bacteriophages, is important for just the next thing here, and its bacteriophages are a type of virus that infects and kills a bacteria. And if we look right here, this is the experiment. This is Hershey and Chase's experiment to determine if it was protein or DNA that was transmitted. So what they did was they used radioactive markers, and they radio-labeled the nucleic acid, the DNA, in one, and they radio-labeled the protein coat in another, and then they had them infect the bacteria and see which was left behind. And they found that the radioactive phosphorus, which labeled the nucleic acid DNA, was left behind. There was no radioactive sulfur, which marked the protein left behind. So they knew it was DNA was transferred, not protein. So here's the information on Hershey and Chase. Concluded that the genetic material of the bacteriophages that infect the bacteria was DNA, not protein. So now scientists knew that genes were made of protein. So this slide right here is extremely important. It's showing the nucleotide bases for DNA. We can see that those blue dots are phosphate groups. Those gray pentagons are the deoxyribose sugar. And then we see those colored shapes are the nitrogenous bases. We have the two purines, adenine and guanine, and then we have the two pyrimidines, cytosine and thymine, and we see that adenine and guanine are larger, so purines are larger than the pyrimidines. Okay, you can remember it. G and A is Georgia. Georgia is larger than Connecticut. Things are pure in Georgia. Okay, CT is Connecticut, GA is Georgia. So you can remember guanine and adenine, G and A, purines larger than CT, Connecticut, cytosine and thymine. Okay? Base pairing, the three parts, the 5-carbon sugar called the deoxyribose, the phosphate group, and a nitrogenous base. So the nitrogenous bases are the purines and the pyrimidines, so adenine and guanine, and cytosine and thymine. Now Chargraph was a scientist who discovered that the percentages of guanine and cytosine were almost equal, and that adenine and thymine were almost equal. So when you're making the DNA double helix, you're going to pair a purine with a pyrimidine every time, the big one with a small one, so that the DNA remains the same shape throughout. And the way it's going to pair up is G with C and A with T. Okay, and we can just shorten the adenine and thymine to A and T, and the guanine and cytosine to G and C. Now Rosalind Franklin used an X-ray diffraction picture technique to get the information about the structure of DNA. Using that data, they were able to determine that DNA was coiled. So we had some coiled DNA, and Rosalind Franklin determined that using her X-ray diffraction technique and reconciling Chargaff's rule and Rosalind Franklin's picture where Watson and Crick, their model of the DNA double helix had two strands wound around each other, and they explained it with base pairing, which explained Chargaff's rule, knowing that A pairs with T and G pairs with C, and each of these bases are paired with each other. All right, and that's how Watson and Crick brought it all together.